In many disorders, including autoimmune diseases, therapeutic antibodies have been established as a successful treatment option. Therapeutic antibodies consist of 3 fragments: the constant Fc fragment and 2 symmetrical Fab fragments containing the idiotypes, the binding sites of the therapeutic antibodies. Their design enables the therapeutic antibodies to specifically bind to a target substance, making them an effective treatment option. However, there is also the possibility of treatment failure. Often the exact causes of treatment failure remain unknown. However, one potential cause, immunogenicity, has recently gained prominence. Immunogenicity is defined as the ability of a substance to provoke an immune response. Provoked immune responses can occur intentionally as well as unintentionally. Vaccines, for example, intentionally stimulate the immune system to produce antibodies against viruses and bacteria. By contrast, unintentional, undesired immunogenicity can also occur, as with therapeutic antibodies. The immune system can initiate the production of antibodies directed against the therapeutic antibody drug. These antibodies formed against the drug are called anti-drug antibodies. The binding of the anti-drug antibodies to the drug results in the formation of a drug-antibody complex. If the binding site of the antibody drug is affected, the target substance can no longer be bound. Those antibodies that block the idiotype of antibody drugs are called neutralizing antibodies and may be a reason for the reduced or absent drug levels in the serum that can be observed. The drug is no longer present and can, naturally, no longer work. Patients who develop anti-drug antibodies may have reduced chances of reaching remission. In addition, patients with anti-drug antibodies may drop out of therapy more often due to treatment failure than patients without anti-drug antibodies. To compensate for treatment failure caused by the reduced or absent serum level of the therapeutic antibodies, practitioners sometimes increase the drug's dose or the frequency of its administration. This, of course, could drive up costs while offering no guarantee of better patient outcomes. Furthermore, the development of anti-drug antibodies can give rise to safety concerns, for instance, an increased risk of infusion reactions. Registration authorities such as the FDA and the EMA therefore demand the provision of immunogenicity data for biologicals and biosimilars prior to licensing. To sum up, treating patients with therapeutic antibodies can result in immunogenicity, leading to several possible effects, such as a reduction in the efficacy of these drugs, increased treatment costs, and compromised safety for patients.